Development of a microfluidic research device to investigate cancer metastasis. Metastasis is the process through which cancer cells from a primary tumor enter the bloodstream and form secondary tumors at distant sites. This is how cancer spreads. Metastasis is a major predictor of morbidity and mortality in cancer patients. This process begins with an oncogenic transformation event, followed by angiogenesis. The tumor then invades the surrounding connective tissue and causes changes in the extracellular matrix and adaptation of the tumor microenvironment. Cells intravasate or enter the bloodstream and become circulating tumor cells. Most of these are rapidly destroyed by the immune system, but surviving cells can arrest in the capillary beds of organs or other tissues. They then extravasate or enter the organ, and a metastatic site is established as tumor cells grow and proliferate. But the details of this process are poorly understood. So we devised a method to study metastasis that attempts to combine the physiologically relevant data we can generate with in vivo models with the high throughput, rapid, low-cost nature of many in vitro methods. Our experimental platform is a microfluidic device composed of three main parts. The first is a valveless pump that reproduces peristaltic flow throughout the system that will provide nutrient and oxygen-rich media, kind of like the heart. The second are tissue channel interfaces, one well for cancerous tissue and one for healthy tissue that might serve as a secondary tumor site. These features alone allow us to conduct studies involving transformation at the primary tumor site and the process of forming a secondary tumor. But circulating tumor cells are a valuable type of cell to study because the understanding of genetic changes that allow for circulating tumor cells to enter the circulation and spread cancer to a secondary site are great targets for novel drugs and therapeutics. With this in mind, we've included a dielectrophoresis sorter to allow circulating tumor cells to be separated from the primary blood flow without the use of antibodies or other methods that may impede our ability to do downstream analysis. Together, the system allows us to study metastasis, circulating tumor cells, and even potentially to do high-throughput drug screening. For the purposes of this project, we chose to focus on a specific experiment. We seed one experimental chamber with NDA MB231 breast cancer cells, an aggressive triple-negative form of breast cancer, and the other chamber contains healthy brain tissue, separated from the circulation by a simulated blood-brain barrier. This serves as a potential metastatic site.
So this is COMSOL, multiphysics software. We use this to modify an existing dielectrophoresis sorter to isolate circulating tumor cells from the blood. Dielectrophoresis, it's an electrokinetic phenomenon where a non-uniform electric field exerts forces on dielectric particles. CTCs have their own dielectric properties, which allows them to be separated from the rest of the bloodstream once dielectric forces are exerted on them. The basic idea is we have these inlets. We would pass through a sample bloodstream sample, and then it would pass through this non-uniform electric field. The forces would be exerted on the particles, and we would have the CTCs separated from the rest of the bloodstream, and this would allow us to isolate the CTCs without needing extraneous cellular markers, and it would allow us for more accurate downstream analysis. We did this with 18 parameters that we got from various literature searches, which are included in our write-up. We have three different physics interfaces. We have the electric current interface, which is to model the electric field in the sorter. We have the creeping flow interface to model fluid flow. We have the particle tracing interface to model the trajectories of CTCs and red blood cells when the dielectric phoretic forces are applied to the particles. We also have three different studies. We have our first study, which is a non-time-dependent study, and we use this to solve for fluid velocity, pressure, and AC electric potential. We have our second study, which is a time-dependent study, which we see the trajectory of the particles with no dielectric phoretic forces. And then we have the third study, which is to compute the particle trajectory once dielectric phoretic forces are exerted on the particles. COMSOL is a fairly complicated software to use. There's a lot of different parameters. So we took a lot of time to do literature searches to make sure that we were able to make sure that we had the right values for the properties that we used.
So for the synthetic data for the dielectric phoresis sorter, I went to the literature and I found data points that they used in the actual experiments. And then based on that, I created a neural distribution with the mean centered around those data points. And for each parameter, I sampled a random value from those data points. I put that all together to make a CSV and exported it for my machine learning algorithm. So for the machine learning part of the dielectric phoresis sorter, the analysis of the data, I did a linear regression with gradient descent. And basically all that means is that my algorithm tries to make a prediction on what the result is or the output is based off of the inputs and the parameter. And based off that prediction and the actual data, it creates a loss function, and the loss function is slowly minimized as the parameters as the algorithm changes the parameters. So here, see as the iterations continues to increase and the parameters change weights, the loss decreases. And from here you have the parameters weights as well as my error, which is just a mean squared error.
To make a simpler model, we choose this like a really simple like a differential equation based like a system that only contain like three that we only focus like three parameter: the angiogenesis and the permeability and the immune system level. Like basically like each like all this equation have like a simple relationship that describe how these three parameters will impact the level of circulating tumor cell and the metastasized like a tumor cell. So like we run the simulation, randomly select those parameters from this range to basically simulate what the real patient, the parameter of the real patient will have. Then we do like a 1,000 trial and get like a metastasis level recording from the end of the 300 discrete stat simulation and add a Gaussian noise. And then we categorize it as like a low, mid, severe metastasis. Finally, we basically plot them to basically show that indeed they are like structural and we could visualize it. So like if we have more time, we will add more parameter and we won't be able to visualize the entire structure. Now we apply machine learning. Since this is a multi-class, three-class classifier, we have to use three boosting algorithms. So each algorithm is a binary classifier. I choose like the first one to like output alpha 1 and DT cell 1 to basically classify the low from the rest. This one mid from the rest. This one high from the rest. And then we after getting trained by the 80% of the synthesized data, we perform testing on the remaining 20%. We include the parameter and we can pass them through both suites boosting algorithm, and we choose the one with the highest margin. The higher the margin, the more confident it get. So and then we basically a max margin, and then we compare it with the true testing label. So now we could plot our training and testing error based on the Raunaq boosting. So let's go. So first we could visualize the result here. Indeed there are structure to this data. We could move it around. And then in reality if we have more time, this will be a multi-dimensional, 10th dimension. And here we show that as only with 100 trial, the training error is around 5%, the testing error is around 10%, which far better than our last attempt, which using the nearest neighbor method. It's worked terribly. So overall, this model have couldn't really represent what true patient is, but we show that it could eventually if we spend more time in learning about microfluidics. And our idea is once we design this sophisticated model in a microfluidic device, we make hundreds, thousands of them. We model the data and we pass it through the learner. Then we could basically gather those parameters from a true patient and predict their metastasis level. This will help a lot in the diagnosis